Hey everyone, today is a very, very short lesson, but make sure you're still paying attention because I feel like this lesson has more information that you really haven't learned yet or before. So let's go ahead and get started. Now that we've kind of talked about arteries, we've talked about veins, we've talked about how blood pressure runs through veins, we need to mention how capillary exchange happens where they release O2, they pick up carbon dioxide, CO2, they give off nutrients as well and pick up waste. The reason we're talking about it now after we talked about blood pressure is because capillary exchange actually occurs due to the different pressures within these capillary blood vessels. So what exactly are capillaries again? Again, they're an intricate network of blood vessels among the body's cells. And no substances has to diffuse very far to enter or leave the cell in order to get into the capillary. What happens are substances diffuse across something called an interstitial fluid, and this is known as tissue fluid. This is going to be found between the cells, hence inter. What happens is these cells release waste or they release CO2. They travel through this interstitial fluid, and then they move into the blood vessel through the capillary walls. This exchange happens because there is a pressure gradient, which is why we talked about blood pressure first. Because of these pressure gradients, oxygen and nutrients tend to leave the blood and move into the tissue cells so that they can get oxygen for cellular respiration and they can have nutrients in order to have energy. And then they also, carbon dioxide and waste leave the tissues and they enter into the blood so that the blood can travel to your lungs and they can release carbon dioxide. Blood also gets filtered by your kidneys, which would help get rid of a lot of your waste that you have from cellular respiration. The route that substances take to enter and leave the blood differs depending on what type of compound is trying to enter and leave. For example, the first method, direct diffusion through membranes, these are usually lipid-soluble substances, so they can go through that phospholipid membrane. These are things like oxygen and carbon dioxide. This doesn't require energy to transfer them across a cell membrane. So direct diffusion doesn't require any energy. They just flow right through. Then we have diffusion through intercellular clefts. These are gaps between cells in the capillary wall, so things can go through there as well. The third way is diffusion through pores of fenestrated capillaries. Now, fenestrated capillaries, these are very unique capillaries that allow exchange of larger molecules. They're pretty permeable, so things that are very big that can't really fit through the membrane itself or through these little gaps, these little clefts, can fit through these fenestrated capillaries that have a little bit bigger pores. These are usually found in your intestine, your endocrine gland, and your kidneys, where they are trying to filter out waste that may be bigger or take in nutrients that are bigger. And the last one is transport via vesicles. These are going to be liquid insoluble substances. These are things that can't really pass through the cell membrane. These things are going to undergo process known as exocytosis. Exocytosis means that it's going to exit. The vesicles are going to leave the cell. And then you have endocytosis, and endo means enter. So these are when things are going to be brought in. Any substances that are unable to pass of one of these four ways are actually prevented from leaving or entering the capillaries. They stay in the capillaries. You don't let them leave. These are things like blood cells. You don't want your erythrocytes, which are red blood cells, and your white blood cells to really leave. The other things is your protein molecules, so blood proteins. If you were going to ask me a question, what type of blood proteins, these might be proteins that actually help your blood clot. You don't want to release those. Okay. So here's a picture that kind of illustrates these 4 different routes of capillary transport. Again, the first one is directly through the membrane, so they just go right through it. The next one is through these intercellular clefts. So again, these are just little holes or pores where they can come into the tissue. The next one is diffusion through a pore, which is your fenestrated capillary. So you can see how much bigger this pore is than this little cleft hole. So this again allows larger molecules. And the last one is if you have a vesicle. So remember, vesicles are these little dots here. They bind with your cell membrane and they can open and release things. Or here, this will pinch off and close and bring it into the capillary. So how does this actually occur? Well, fluid movement out or of or into capillaries is going to depend on pressure between the difference between 2 pressures. And this is why we talked about blood pressure first. The first one is blood pressure, and this actually forces fluids and solutes out of the capillaries. And the second one is osmotic pressure. This draws fluid into the capillaries because blood has high solute concentration due to the plasma proteins in it than the interstitial. And one thing to know about fluids is going to go wherever the solutes are. So if there's high solute versus low solute, fluid is always going to go wherever there's high solute. I always think if they try to dilute it. So as a rule, blood pressure is typically higher than osmotic pressure at the arterial end. Think about what we know about arteries and how blood pressure works. Remember, blood pressure works along a descending gradient. So, of course, it makes sense why it would be higher at the arterial end. And then on the opposite end of that spectrum, blood pressure is lower at the venous end. And think about what we know about veins. Veins have lower pressure. The reason this is, and the reason why this is very important to have it this way, is if blood pressure is lower than osmotic at the venous end, fluid will move out of the capillary at the beginning of the bed, and it's going to be reclaimed on the opposite. So it can take out that fluid, it can do its little gas exchange and nutrients exchange, and then give it back. Now, not all fluid that is exchanged between your capillary bed and your tissue is reclaimed. Some of that fluid gets left in that tissue. You get pooling fluid, and that's usually taken care of by your lymphatic system. So over here, we have this little chart. Up here, we have your tissue cell, your interstitial fluid between the cells. Down here, we have the capillary. So here we have the arterial end. Here we have the venule end. When it enters, again, fluid is going to move out. Here, when you get to the venule end, it's going to move in. And they give little examples over here. At the arterial end, blood pressure is more than osmotic, so fluid goes out. Here, blood pressure is less than osmotic, so fluid is going to go into the capillary. And that's the end of the lesson today. Let me know if you have any questions, and I'll be more than happy to help. Have a good day.